Hi everybody. Today we are going to study antibiotics. In this topic, you will learn about the structure of the bacterium, how antibiotics work, why antibiotics don't affect the viruses, antibiotic resistance, what is vertical gene transmission and the horizontal gene transmission, concepts of antibiotic resistance. There are some keywords that help to us. These are the antibiotics, osmotic lysis, peptidoglycan, or another name murine, autolysins, plasmid, superbug. Firstly, let's look at the structure of the bacteria. Generally, all the bacteria are divided into two main groups according to their gram staining. These are gram-positive bacteria and gram-negative bacteria. And the bacteria are prokaryotic cells. They have no nucleus, and some membrane-bound organelles are also absent. They have cytoplasm, genetic material, DNA. Also, some organelles are present in cytoplasm. Outer part of the cell covered with the cell membrane or plasma membrane. And then after the plasma membrane, they have cell wall. This cell wall is made up of peptidoglycan, or another name, murine. And as you see in gram-positive bacteria, the cell wall is thicker than the gram-negative bacteria. And out of the gram-negative bacteria also present the outer membrane. This is a general structure of the bacteria. And what are antibiotics? How the antibiotics work? Now, antibiotics, these are the chemical substances produced by the microorganisms, and they destroy the other organisms or inhibit their growth. By this, they kill the microorganisms. And antibiotics work by two ways. The first one is interfering with the cell wall synthesis. It broke down the synthesis of the cell wall. And second one is making pores on the membrane of the bacteria. And look at here, in bacterial cells, water always continuously enter by osmosis, entering of the bacteria to cell cause swelling and the burst normally. This is called osmotic lysis. And osmotic lysis doesn't occur in bacterium because it has a cell wall and this cell wall prevents burst like a plant cell wall. And the difference of this bacterial cell wall from plant cell wall, it's made up of peptidoglycan instead of the cellulose. When water enters bacteria by osmosis, the peptidoglycan pushes against the cell wall. This prevents further entry of the water. Peptidoglycan are made of amino acids and the sugars. Now, these are long chains. These long molecules held together by the short peptide molecules that form the cross-linkage. Now, as you see, here are some cross-linkages are shown. And in young bacteria, when they secrete enzymes, these enzymes break down this peptidoglycan cell wall or these peptide bonds. And then it forms it forms tiny holes on the on this peptidoglycan cell wall, and the hole allows the cell wall to stretch, and then they grow. After that, again, these holes are filled with the new peptidoglycan cell wall. Now, in this picture, shown the structure of the bacteria and some antibiotics and their effects. Now, here are groups of the antibiotics, for example, penicillins, bacitracin. These antibiotics inhibit the cell wall synthesis. Or there are another group of the antibiotics, erythromycin, tetracyclines. This group of the antibiotics inhibit the protein synthesis in bacteria. Some of them inhibit the nucleic acid replication. Some of them destroy the plasma membrane. Some of them inhibit the synthesis of the essential metabolites. In this group of antibiotics, we will focus on the penicillin. Now, look at here, how does the penicillin work? Now, we have talked about this. The cell wall is made up of peptidoglycan, and peptidoglycan, when you look at the structure of the peptidoglycan, it's made up of amino acids and sugars, which held together by peptide molecules. And penicillin, how this penicillin affect on the cell wall formation? Now, it inhibits some enzymes that synthesize these peptide cross linkages. If peptide cross linkages are absent, the peptidoglycan cell wall weakens, and then it causes ruptures. After that, the cell will burst. And look at here, sometimes when we ill, we use antibiotics, but they have no any effect on our body. For example, if we have a flu, and if we use some antibiotics, it has no any effect on the flu. Why? Because some disease come with the viruses, not with the bacteria, and antibiotics don't affect viruses because there are some reasons for this. One of them is that viruses don't have own metabolism. Always they are found in host cells. And another one, antibiotics act on organelles that are absent in viruses. Third, viruses have a protein coat rather than a cell wall. Fourth one, viruses live inside the cells of the host out of reach of the antibiotics. That's why the antibiotics don't have any effect on viral disease. Now, how antibiotic resistance happens? Now, when we use the antibiotics, it increases the chance of mutation in bacteria. And due to this, some mutated bacteria build up a tolerance to antibiotics. In this case of resistance to penicillin, mutation results in a certain bacteria able to make a new enzyme that broke down a penicillin called penicillinase. Mostly, mutation occur randomly, very rare. Not all mutations are useful. Mostly they are harmful. Harmful mutation cause death of bacteria. And this gene coding for penicillinase pass from one generation to the next generation with vertical gene transmission. Now in here, the one cell divides into two by the binary fission, like this. after some period in a population, the number of the new mutated bacteria will increase. The allele for antibiotics resistance can be carried on the plasmids. Plasmid can be transferred from one cell to next with the here in the long term the frequency of allele for penicillin resistance increase in a population, and the allele for the resistance can be carried on these plasmids. Plasmids can be transferred from one generation to the another generation with this plasmid. And there's a two alternative way of the gene transmission. One of them vertical gene transmission, second one not the horizontal gene transmission. And in a horizontal gene transmission, mutated bacteria able to make conjugation with another normal bacteria, and in here the plasmid of the antibiotic resistance pass to the another cell, and then both of them now look at here, look at this picture. In this, both bacteria cells have what? Antibiotic resistance. Now, this process called conjugation. Through the conjugation, plasmid pass to the next cell. And here's an example for this. Now, here's a Escherichia coli bacteria. It has own DNA. In this DNA, some part of DNA is a gene for against resistance to some antibiotic, and with the conjugation, this antibiotic resistance gene can pass to the another bacteria. Here's a Streptococcus pneumoniae, which cause disease on our upper respiratory canal. As see here, after the conjugation, both of them present antibiotic resistance gene. And this horizontal, gene transmission can lead to certain bacteria accumulating DNA plasmids. For example, these bacteria may be able to take many different kind of antibiotic resistance gene from other bacteria, and it causes accumulation of DNA plasmids. And this gives this bacteria resistance to a range of antibiotics. And there are multi-drug resistant bacteria, and these are called as a superbugs. And in a superbug, the bacteria able to resist several different kind of antibiotics. Now, some countries on the world face crisis about superbugs. The first three countries which are face this crisis: India, China, and USA. These are faced with superbug risk. And how we reduce antibiotic resistance? There are some ways to reduce the antibiotic resistance. Now, firstly, limit antibiotic treatments in minor illnesses. If the symptoms are not serious, you may not use the antibiotic. And second, encourage patients to always complete the course of antibiotics as prescribed, even if you are cured. You should complete the course of antibiotic course. to prevent patients from building up a supply of unused antibiotics from previous prescriptions and then using them later in small doses than they should. Avoid the use of antibiotics in treatment of minor illnesses in domesticated animals, for example, for example, cats or dog, which are found at home. Limit antibiotics use in the prevention of the disease among intensively reared animals such as chickens. Limit the use of antibiotics by farmers and the companies to reduce disease and hence increase the productivity of the animals. It's also important. Hold back certain antibiotics for future use when others have been made ineffective by resistance. Now we have finished our topic. Let's pass the review questions. Now, what are antibiotics? Now, antibiotics are the organic substance which are produced by the bacteria or other organisms, and they are these antibiotics can be used to kill the other microorganisms, especially in patients who have serious disease. Now, how penicillin affect harmful bacteria? Penicillin affect the bacteria with the breaking down of the peptide molecules in their cell wall, peptidoglycan cell wall. What is penicillinase? Now, penicillinase, it's an enzyme, penicillinase. Now, some bacteria are resistant to penicillin because they have because they have what? Penicillinase enzyme. This penicillinase enzyme broke down the penicillin molecules. And because of this, some patients cannot cure with using the penicillin. Why antibiotics don't work when we have flu? Now, flu is a viral disease, and antibiotics have no any effect on this flu. Look at the name also now, antibiotics, anti against to bio living things now. That's why it has no any effect on the flu or on the any kind of viral disease. What is vertical gene transmission? Now, vertical gene transmission, it's a passing of the gene when the cells divide to new cells. What is superbug? Now, superbug, some bacteria have resistance to against so many different kind of antibiotics, and at this time, if these superbugs are harmful and cause disease on organisms, we cannot cure with using the several antibiotics. It's a really big risk to cure some patients. Our lesson has finished. You have homework on EduPage. See you